Every year, the College of Veterinary Medicine at Kansas State University presents to one of its outstanding faculty the E.R. Frank Award. E.R. Frank, a longtime faculty member at Kansas State in equine surgery, and this recognition is presented to an individual who displays the same professional essence of Dr. Frank as he succeeded in his career. Well, the award winner this year joins us now. He is Dr. Deryl Troyer. Deryl, congratulations, first of all, for this recognition.
Thank you very much, Eric.
And you have spent over 40 years off and on here at Kansas State, have you not?
I guess that's about roughly the number of years, yes.
And it's been back and forth from K-State into career paths and then back again. But let's go back to the origin. You're a native Nebraskan, correct?
Correct.
Born where?
Well, it's pretty close to Geneva, Nebraska. That's the closest major town, little town south of York, Nebraska. So it's kind of the toward the eastern part of the state, southern edge, about 30 miles from the Kansas border. So I'm almost a native Kansan. So.
Were you raised in an agricultural background there?
Yes, grew up on a dairy farm.
Did you have inclinations early on as far as animal health then, while you were growing up?
That's really the primary reason I got interested in veterinary medicine. I really wanted to get into a dairy type health program of practice, and fortunately later on I was able to do that to some extent. So, but that was my major early interest, was the dairy production medicine.
And you came to Kansas State, what, in the late 60s?
Yes, I started in veterinary school, I think, in 1968, and I graduated in 1972.
With designs on a practice in dairy health, or were you broadening your horizons?
Well, initially, and this is often the case, we see this with our students very frequently. You start out with a certain idea, and then for one reason or another, you end up in a different sort of a practice setting. And I actually went into a small animal practice in a suburb of Chicago initially, and I just thought I'd better try that, or if I didn't try it, I would never try it again. And I really loved the practice. My wife and I just didn't particularly like living in an urban environment, so that kind of that's the reason I really didn't pursue the small animal.
So after a brief stint in that practice, you were lured back to Kansas State, right?
Well, I actually practiced in a mixed practice in southeast Nebraska, in Pawnee City, Nebraska, for 8 years, and a partnership, and which I enjoyed a lot. We actually did do a fair amount of dairy contract work, and then I had the opportunity to come back to graduate school in Kansas State, and I finished a PhD degree in 1985, and from there I took a job on the faculty at the University of Illinois. I taught a course called histology, embryology, and did some research there, and then luckily this position opened at K-State, and I came back on a faculty here.
And when you came back, your specialty was in physiology and pathology, or more than that? What?
At that time, I was very much interested in molecular genetics, and specifically, I was involved with developing gene maps for livestock species, and I actually worked in that field for probably 10 years, somewhere 10, 12 years, and the gene maps eventually got developed, and once the gene maps are developed, there's really no more gene mapping to do. So that's where I shifted gears a bit, and my collaborator, my very good friend Dwayne Davis, and I over in animal science just got the idea that we could isolate cells from the umbilical cord. And we had been working with bone marrow stromal cells before that, and so we were really thinking about other potential sources, and so that's opened a whole new area. So
elaborate on that, for that's one of the main hallmarks of your career here at Kansas State, what that has led to.
It's developed into a worldwide projects that involve these cells, and they're mainly human cells. We actually started out working with cells from domestic species, and about 8 months after we started working with the human Wharton's jelly cells. And you know, it's just astonishing to read reports frequently coming from labs all over the world. And these cells have remarkable ability to form a variety of tissues. So they're being pursued for their ability to regenerate for bone or cartilage or heart or brain or other tissue regeneration. The most exciting thing to develop, and this has been probably in the last 5 years, doesn't involve work, or at least my lab right now, but it's been shown that these cells can dampen the immune response. They can have an immunomodulatory role, and that's the most exciting thing really that's come along. And clinical trials, these are human clinical trials, and this is published work. These cells have virtually saved the lives of kids undergoing something known as graft versus host disease, and these kids were actually at grade 4 and steroid resistant, and the prognosis almost is very, very grave. And so that is the biggest thrill I think that I could possibly ever have imagined, to read the results of a clinical trial where lives are being saved by something that otherwise wouldn't have been available. And I might point out that they are similar to other related stem cells that can be isolated from adult tissues, like bone marrow, adipose-derived mesenchymal stem cells. But they appear to, for whatever reason, have a greater effect. And you know, the sky is really the limit. And I think that someday they're going to be very famous cells. So.
Way back when, did you ever imagine that your work would transcend animal health into human health at that magnitude?
I think, well, I should point out, the first glimpse, one of the first glimpses was that these cells can actually inhibit fibrosis. And that's been one of the really, really fantastic spin-offs of not only these cells, but other cells. They not only have the ability to regenerate damaged tissues, but they do other things. For example, they might secrete nourishing factors that help the resident stem cells in the tissue do what they're supposed to do. They might dampen immune effects. And in the case I'm referring to, they have been shown to basically eliminate fibrotic scar formation, in tissues like the lung or the liver cirrhosis. This was actually a liver cirrhosis model. And so that's a huge unmet need, and that's another reason these cells are very exciting. So I didn't really, you know, when we started, we were, and they still have, I might add that their potential impact in veterinary medicine and agriculture is still very much there. In fact, Mike Dwayne Davis and I have shown that if you give these cells to newly born piglets, give them orally, they will actually cross the lining of the gut, and they will engraft in the connective tissue lining. And we don't know how we'll use this yet, but it's just an amazing finding. You can give these things to a newborn animal, they will actually, you know, traverse the epithelial lining and set up residence. So that's another example. And so maybe in veterinary medicine or agriculture, these cells might someday be an off-the-shelf source of cells that might be engineered to make an antimicrobial peptide, you know, to combat antibiotic resistance, or they might be used to develop a protein that can serve as a vaccine or something to that effect. And we think they might have a lot of ability to enhance wound healing in domestic species. So
It's worth noting as well, you're aligned with research work at cancer centers at Kansas State University, at the University of Kansas. So this dovetails well, doesn't it?
Absolutely. And the way we're using these cells, it illustrates again the idea that they can do things you normally didn't really envision them doing. And in this case, we are using them as sort of stealth delivery vehicles to specifically target cancer. And we can either engineer these cells to secrete a protein that works a lot better if it's secreted locally within a tumor, or we can preload the cells with magnetic nanoparticles or other kinds of nanomaterials, often carrying a therapeutic payload. And we let those cells then ferry this therapeutic payload directly into the tumor and hopefully avoid a lot of the side effects that take place.
Really is exciting work, isn't it? You do want to acknowledge a number of people that you've worked with over the years here at Kansas State. You mentioned Dr. Dwayne Davis out of the Animal Sciences Industry Department as one, but there are numerous others you think are deserving of a mention too.
Oh, absolutely. Of course, Dwayne is just fantastic. He's been a wonderful, wonderful collaborator. And I'd like to just acknowledge some KSU CVM alumni that have had a really profound impact on my professional development. And the first name that comes to mind is a superstar among K-State alumni, Med alumni, and that's Jim Kaufman, Dr. Jim Kaufman. And I really got to know Dr. Kaufman very well after he had retired as provost, and he helped us launch our Midwest Stem Cell Institute. And what a talent for galvanizing people and ideas and just overcoming obstacles. So I really appreciate that. And I'd like to acknowledge Dr. Russ Fry, who was my first department head when I first joined the faculty here. And Dr. Fry was just a huge inspiration. My colleagues in the Department of Anatomy and Physiology, particularly in the anatomy section, Dr. Wally Cash and Dr. Judy Klimek, and we've just had a really good working relationship in the teaching aspect, and that's been a wonderful team. So
there's so much to your story, we could spend the entire day on it, but let's round this out by having you acknowledge what this recognition means to you, because you're very familiar with the work of Dr. E.R. Frank.
I think the biggest reason that I am deeply honored is because Dr. Frank was such he was so far ahead of the field and such an innovator, and just even be mentioned in the same breath with Dr. Frank's name is an honor, and that's I think that's the most exciting thing about what we do. We find ourselves often in some uncomfortable terrain where we're investigating some new paradigm that we really don't have any guidelines, any milestones to follow. But and that's what Dr. Frank did. He had to do the same thing. He generated new ideas, and then he set about testing them, and so I'm sure he went through the same process.
Well, hearty congratulations to you for this recognition, Deryl.
Thank you very much.
Thank you for sparing a few moments.
Appreciate it.
He is the recipient of the 2012 E.R. Frank Award for the Outstanding Faculty in the College of Veterinary Medicine at Kansas State University, Dr. Deryl Troyer.